A very warm welcome to all the participants who have joined us from all over the world. Welcome to this 9th session of BISMiS Live, our monthly seminar series, which hosts microbiologists and taxonomists who are working with bacteria, archaea, and microscopic organisms. I'd like to briefly introduce our speaker for today, Professor Ramon Rossello-Mora. He'll be speaking on lessons learned from the LTP that reinforces the robustness of the 16S as a phylogenetic marker for biodiversity studies. Ramon has been an excellent contributor to the society and to the microbial systematics field in general. He's currently a senior researcher at the Spanish Council and working in the Joint Research Institute leading the Marine Microbial Group. He did his PhD at the University of Balearic Islands and a postdoc at the Technical University of Berlin, then Technical University of Munich, and the Max Planck Institute for Marine Microbiology in Bremen. He specialized in molecular microbial ecology of marine sediments and hypersaline environments, as well as in theoretical and practical aspects of microbial taxonomy. As a taxonomist, he is very well rewarded and very well known in the field. He's been granted with the 2017 Burgess Award, and he is the Vice Chair of the Judicial Commission of the ICSP. And he's also the Executive Editor of the Systematic and Applied Microbiology. He's a member of the European Academy of Microbiology since 2016. He is an expert on microbial systematics, taxonomy, phylogeny, molecular ecology of marine and hyperhalophilic prokaryotes. And he's been working on genomics and metagenomics of prokaryotes, as well as different aspects of molecular microbial ecology. So with this brief introduction, I welcome Professor Ramon Rossello-Mora, or Ramon as we fondly call him. Ramon, please, you can take the stage and begin your presentation. And thank you for joining us.
Thank you very much, Kamlesh, for the invitation. Thank you very much, the BISMiS, and I'm really impressed by your capability to organize these meetings. It's a very important challenge for taxonomy. My talk, actually, I want to talk about 16S rRNA because it's something that in the last very important meeting talks that I could listen, this has been given a little bit of attention, not much attention. This is my group. One moment. So this is, okay, so sorry. I wanted to use the laser, but it's not working for me. The laser, yes. So this is my group. As you can see, it's a very dynamic and a very young group with whom we are working. And in case that you want to follow our information, you can just follow the different Twitter accounts. This talk has not been possible without the help and the work together with Rudi Ammann and Wolfgang Ludwig, and also from the team of the LTP, who has been working intensively in the last days and year. The IMEDEA, where I work, actually, this is our institute that is located in, in the island of Mallorca. You can see here is in Spain, in the Western Mediterranean. It is known, Mallorca, for the many parties for the Central European tourists, but it has interesting nature and also interesting culture issues. The institute is located in the mountains, and we are a very interdisciplinary institute where I'm the only microbiologist, but we work with marine sciences. So before starting with the main topic of my talk, which is the 16S rRNA, I want just to introduce an important theoretical aspect, and this is what taxonomy is. Taxonomy is probably the discipline that has been one of the oldest disciplines that we have in biology, because it has been used at almost 300 years. And this discipline is the only one that has had a lot of amateur working on it. They had many different definitions, But the one that actually is more succinct of all of them is the one that was given by Mayer, which is the theory and practice of classifying organisms. All of them, more or less, will explain the same definition. The most, for me, important taxonomist, or actually my most loved taxonomy, that is Cohen, he already indicated that taxonomy is made by three different disciplines. So one is classification, another one is nomenclature, and the third is identification. That is the purpose of the taxonomy. It's the way to identify the organisms that we have and ensure that we understand and we clearly know who they are. He already complained, and this is many years ago, that too much attention had been given to nomenclature, and this is also holding true nowadays, under my opinion. So taxonomy, from my point of view, is something that pursues classification, because classification is used in both the activity and the system that we create. And the general purpose is to have a classification that is universal for any kind of organism. We should create a database that is comprehensive, harboring all the information possible on the nature, so beings. And the system that we create should have several premises that are important. One should be operational. This means that it should work. Should be predictive, and this is very important. When you hear a name, you have to be able to predict properties of this organism, properties like genetics, or properties like phenotype, or ecological properties. And what is more important, this system should be universal, should be embracing all kinds of organisms. And finally, it should be really pragmatic. It cannot be so complex that nobody can use, because the users are not only taxonomists, the users are also any kind of other disciplines in biology. So this system, we are pursuing something that is natural. So we want to just organize a natural system that reflects the natural relationships, but we have to understand that if we want a universal application, this will be somehow artificial, as I can explain to you. So the first categories that we have in taxonomy were described actually by Aristotle around 2,300 years ago, and he devised the first two categories, that are genus and species. They were devised because of other reasons, not because of biological reasons, but these two categories have prevailed until now. It was not until Linnaeus, around 300 years ago, that he created the first hierarchical taxonomy in his work on botanics, and he created three additional categories: the kingdom, class, order, genus, and species, that are still in use nowadays. But the taxonomy was, as I said, full of amateurs, and then this created a very big increase of knowledge, that what the taxonomists could see, especially the Mayer and Simpson were the ones who created the categories. They saw that not all organisms were fitting in all categories devised, so they had to create 14 more categories in order to fit all the different groups of organisms that they could see. If we look at bacteriology, if we look at microbiology, we only use 7 categories. Actually, we use 6 categories up to now, and this year we increased the new category of phylum that was not having standing in the bacteriological code, and luckily it has been included this year. And we will discuss this a little bit later, because this is a controversial. So what I was saying is that in bacteriology, we are only using these 7 different categories, and phylum is the last category that has been included this year because up till now it had not standing in bacteriology. So what we have to understand is that only species is the only category that is made by real individuals, and all other categories are actually artificial constructs that we want to use in order to put together the different groups that we see. So the system, in any case, is artificial. I want also, before we continue with the 16S RNA, to emphasize that we have a difference between what is the concept and what is the definition of species. The concept of species is the idea that we have of what it is. So generally, it should be something that is immutable. And I don't think that anybody will contradict me that we will believe that a species is a monophyletic group, which is this a very important premise, of organisms with high genomic and phenotypic homogeneity. This is very general, but is what we are actually circumscribing. The problem is, and this is where the definition is the way. How do we circumscribe a unit? And this depends on the observable, on the character that we can observe. And these changes, especially in microbiology, depending on the technical developments that we are having. So, as we can discuss now, cultivation has been one of the major bottlenecks in order to describe prokaryote species. So the first premise in order to describe something is that we need to go to the environment and bring the organism into pure culture. This means that this jump is probably the most important premise and also bottleneck for describing the diversity of microorganisms. But once we have in pure culture an organism, then we can apply the best practices in defining and circumscribing this organism. So when we have this, and the best practices are, we have to try to describe that the group of organisms are monophyletic. We have to describe that the group of organisms share a lot of genomic characters, and also the same applies for the phenotype. I will especially only discuss the problem of monophily and how do we concentrate on phylogeny, because it's the subject of my talk. I will not discuss anything related to genomic homogeneity and any RNAi or AI values. The thing is that once we have the organisms described, we must deposit it into a living, as a living culture, into collections. And this is one of the premises of the bacteriological code in order to validly publish the name. And then it goes to the official nomenclature lists. Then, of course, you have to publish the protolog. That is, I guess that most of you already know how it works. You have to publish a protolog in one of the journals that are dealing with taxonomical issues. And if you publish outside the International Journal of Systematic Microbiology and Evolutionary Microbiology, you have to you have to ask the list editor to validate your names. If we look at what has happened in the past and what is happening now, what you can see, I made this survey around 3 years ago. I made this survey, and what you can see is this survey was made in the year 2018, I think, in 2.5 years. And you can see that in the IGSEM, around 87 of the descriptions were based on a single strain, and only 13 were based on more than one strain. And what you can see is that 91% of the description were just aerobic heterotrophic, which probably are the least abundant organisms on the biosphere. And finally, when we check about the taxonomical diversity of these organisms, we can see that they belong to 4 major phyla: Proteobacteria, Bacteroidetes, Actinobacteria, and Firmicutes. Okay, these names have just been changed by Aaron and George currently, but I still use them. But we can discuss this later if you want. I don't think it's worth to discuss because they are well proposed. But the thing is that these four major phyla are the ones that we recover in culture and are the most abundant in our collections and classified. But if we look at the work of Phil Hugenholz, around 20 years ago, what we can see is that not much has changed. He already said that at that time, the real diversity that we got in culture only a partial amount. And from this partial amount, there were four phyla that were being recovered, that they have different names now, but are actually the same kind of organism. So something has to be done. And this is something that has been discussed already here in this frame, because we know that we have a large amount of organisms around there. So Barney, he calculated around 6 to the 30, 4 to 6 to the 30. It might be true. I don't think it's something that is very different from the real and reality. And when we look at the number of species that we have been able to classify up to now, we have just 17,000. And we, in our calculation with Rudy Aman and Pablo Yarza, we think that we have really our estimates between 1 to 10 million. But the most optimistic, they think that we have 1 million of millions of species around there. And so when we look at this contrast in numbers, then we know that we have to do something. And we have now the opportunity that is metagenomics that allow us to understand genomics of organisms that are out there without having to bring them into pure culture. If we just do metagenome surveys, for example, or single cell genome amplification would work also, but the metagenomics, especially now the bioinformatic tools are working very well. They allow us to construct the metagenome metagenome amplified genomes, metagenome assembled genomes, and in a high quality, in such a high quality that the information that we gather, it's enough for not only calculating taxonomy thresholds like the ANAI, also it's important because we can retrieve the phenotype, we can infer phenotype, but we can also do any kind of phylogeny, retrieving 16S rRNA if it's possible, that in some cases works, or using concatenates of genes as other people do in order to understand the genealogy, understand the genomic homogeneity, understand the physiology and ecology, and then finally to be able to classify the organism. This is an example of our work done by Tomeu Vivet that we have been working with Cotyloryza tuberculata, which is jellyfish. And we could, by metagenomics, be able to define different organisms, and we could see those organisms using in situ hybridization techniques in order to be able to locate them, to understand who they are, and to see and also understand their metabolisms. So this is going to work, and this is something that is probably will be one of the major sources of diversity. Unfortunately, as it has been also discussed, these marks and sacs cannot be named formally under the bacteriological code because we cannot really bring them into pure culture. Then something has to be developed, like the SIG code that is under its way. But that said, I want to just concentrate on the 16S rRNA because it's the topic of my talk, and I will concentrate on especially how can we use, or which molecules do we need to use in order to do phylogeny, be able to demonstrate monophily and use it into taxonomy. But in order to understand the real importance or the real relevance of the 16S rRNA, we have to understand also the history of taxonomy. Until the 60s of last century, and when no molecular biology really existed, the classification of organisms was made mainly by phenotypic characters, rudimentary characters. But then the most important point was actually the discovery of the DNA being the genetic encoding information molecule, and this has changed the view and the capabilities of microbiologists to name organisms. So when this was feasible, the first techniques comparing genomic information appeared, like especially first the GC content comparisons, and then the DNA hybridization. And this DNA hybridization has been especially important in circumscribing species along the history of taxonomy since the 60s. And this has been the technique that has been relevant and mostly relevant to circumscribed species, not higher taxa. But then one of the major breakthroughs in microbiology, especially, was when the first RNA could be sequenced. And this, we are talking about not only 16S, but we talk first about 5S RNA. And this has been really influencing a lot. If we look here in blue, in red, we have the number of species that yearly had been classified. And we can see that from the moment of the 16S, the first 16S RNA sequencing, then an increase, an arithmetic increase of number of description of species occurred. Also, what we can see is that you have to see that the scale is different. Here is for species, here is for gene increase in the database. You can see that the 16S RNA increase has followed an exponential increase. And this has been important because during all these years, we have had the 16S RNA ruling the description, especially because many descriptions were based only on 16S RNA uniqueness, and many of them in a single strain species description. But now, when the first whole genome sequences occurred, now we are entering in a new phase of taxonomy in where the whole genome analysis are also ruling the taxonomy. Is in the one side is substituting the DNA hybridization. So this technique is being substituted by whole genome comparisons like NAI analysis, AAI, or any other parameters, but also now it seems that there is a shift on the analysis of the phylogenetic analysis from the 16S RNA to whole genome analysis. And as I will try to explain to you now, it is not mature enough to be used. This is my point of view. So if we look at the history of the 16S RNA, especially of the rRNA in this case, we can see that the first work that was done in the late 70s of last century, it was a very tedious work, especially working with a lot of radioactivity, that they were the cataloging, the RNA cataloging. And with this technique, they could the first device and see that they were the three primary kingdoms or the domains of life. It was not until the first sequencing that it occurred in the late 80s that the 16S RNA could be used for reconstructing phylogenies using the 16S RNA as a molecular clock, and this has been the major breakthrough, I think, in taxonomy of prokaryotes. Then we could include phylogeny into taxonomy, and this has been very important. And why using especially 16S RNA molecule? Why? Because, or especially ribosomal RNA in this case, because they have a natural gene amplification. So at that time where no amplification PCR existed, it was very important because those molecules were highly amplified within the cell. So you could feed the cells with radioactivity, and then you could isolate radioactive ribosomes that could be used for or cataloging or sequencing. Also, because this is a non-protein coding DNA, and this means that each base counts, not like in the codon usage that the third base can somehow freely mutate with silent mutations that do not really influence on the analysis. and on the functionality of the protein. Also, the 16S RNA especially, it happens also for the 23S RNA. It has a secondary structure, very complex secondary structure, full of different domains that have importance in the functionality. And this has been not only important because of the conservation of the gene, but also important because we can make, as I will tell you, a universal alignment that is the most important thing to do when you do phylogeny. And finally, also, you can already also see here, for example, we have a different conservation region. So we have highly variable regions and we have highly conserved regions in the genome that are just not concentrated in one side, but along the whole molecular curves. And you can already see here, this is a picture of the Aman in where the intensity of the dots indicate the conservation state. So those are highly conserved, and here that are very faint, are very low conservation. So you can see that the conservation, highly conserved or highly variable regions, they are just scattered around the gene. And this is very important because the whole gene counts, not only counts a piece of the gene. But the most important usage of the 16S RNA especially is because we have a universal alignment that can be used for all kinds of organisms, and also the secondary structure is useful to improve manually and automatically probably the alignment, because the different pairs are just complementing in different helices, and this helps to improve the position of the homologous bases. To this work, to this purpose, Wolfgang Ludwig, he developed the program ARP around 25 or more years. And this program ARP has been in use and improved along all these years. And now we are on the 7th edition of this ARP that accounts with a lot of different tools that you can download from these different websites. The ARP program package is a very useful. It's a little bit complex, but this is it's complex because of the high operativity of the amount of tools that are included in itself. But during these many years of improvement, you can see that this operativity is very good. It's especially suited for the 16S RNA and the 23S RNA because has been specially used for that, but it's suitable for any kind of protein. We use it for any kind of alignment. And it has a lot of tools that with different algorithms for reconstruction, filters, you can do bootstrapping, and many more features that you can read here. I will not I will not talk about them. You can recall, you can revisit this talk in the future and see all this, or you just go to the website of ARP and check it, because it's really a very useful work package. So what we can see is that no other gene in our universe of genes in sequence has the same database as the 16S RNA. In the 90s, we accounted with around 1,000 sequences, and then 26 years ago, now we might account with around 20 million sequences. Silva, that has been curating this 16S RNA database for many years, in the last release in December 19, they already found 30 million sequences in the database. One has to say that 3.5 million have been discarded because they are either wrong or bad assigned, or they are not 16S RNA. But nowadays we account with around 20 million. Let's say we will need to remove 5 million of them, but still a very large amount of information that no other gene will have. So therefore, this is extremely useful because we have a very vast information that we can use with. But also this tool, this 16S RNA, allow us tools like the the design of phylogenetic probes, for example, like to use for example, in microscopy and in fluorescence microscopy, and apply it to techniques like a microradioautography, like for example here, or stable isotope probing, and many other features. And also it's important to note that we can make a single tree that includes bacteria, archaea, and eukaryotes. For the purpose of making a tool for taxonomists, we developed what we call the all species living tree project. Actually, it's a little bit ambitious, and I think we just talk now about LTP, but the LTP has been initially developed by Pablo Yalza, my PhD student, but with all the years that we have been developing, we have had different curators. Now we are working with Ana Suarez, who is curating the very last versions of the LTP, is whom you have to contact if you need any help. This project has been developing in the last 14 years, and the purpose was actually selecting the adequate sequence, type strain sequence, for taxonomic purposes. We found a lot of errors in the databases so that we could curate this, and it was a hard work because many sequences, as you can see here, could be not used because of errors, but we curated this. But one of the achievements that we also did was at that time, with a collaboration with a very large amount of different strain collections, we could finalize the whole catalog of 16S RNA of all classified species. Of course, not all could be sequenced because many of them were unculturable, and many of them, they did not have a culture deposited, or it was lost. But at the end, we could finish with the complete catalog, which was an important achievement. But an important achievement is also to have a use of this gene sequence in taxonomy. And one of the important things is to find thresholds. And for the 16S RNA, Erko Stakeland, he has been making a lot of thoughts. And first, he could set a threshold of 97%, then he rose it to 98.7. That means that everything that is lower of identity lower than this value do not need to be DNA analyzed. So this means that two organisms that have below this value of identity are most probably different species generally. But if you are above, you should prove it using a genome-to-genome comparison analysis. It doesn't mean that it belongs to the same species. It means that you need to prove it with whole genome. But nothing has been evaluated for higher taxa, or had been evaluating at that time when we thought we did the work. Only the genera. There was an unwritten rule that it was spread among the taxonomies that 94% could be a good threshold. And this has been perpetuated, and this has been influencing the way that we circumscribe this category. So what we did, it was when we had this highly curated database, we tried to understand how taxonomies were classifying the categories in order to find threshold that could be used for the future. So we used the best number of species, the best number of groups that had at least 3 representatives in it, and then we came to these different minimum categories. So the species we didn't, this is the one that Erko was using, but then we devised the different values in order to have threshold for genera for in higher taxa. So those are the minimal values that taxonomies have been used, actually not used. The taxonomies have been circumscribing these categories. And phylum, that is a category that had not standing until now, is a different case that I will explain to you in brief. But the thing is that you can see that the lower that we go, the broader is, so the higher that we go in the categories, the broader it is the variability in what taxonomies they did. But this is because the number of categories is lower and because we did not have really rules. But these are the thresholds that we can use. It was important for us in order to check the scroll, the tree of life that we had, in order to be able to distinguish different groups of organisms that actually belong to different categories, but they were understood as the same. For example, it was very normal that microbial ecologists compare the SAR11 together with the Rosseobacter clade, but you cannot really compare because Rosseobacter is probably one family within Rhodobacterales, and SAR11 was one division with one phylum with 3 classes, 5 families, and 5 genera. So these use of thresholds are important because it can help. Another thing that we proposed in the work of Pablo Yarza the work of Pablo Yarza in 2014 is that was not successful. We proposed at that time already a nomenclature system for the uncultured in where we could categorize different groups that we saw in different families and higher taxa, but we could not convince Silva to implement it, unfortunately, and then this proposal has been forgotten. This proposal is not very far away from what Phil Hohenholz yeah, they have proposed. So at that time, in 2012, we wanted to calculate how many species were we seeing in our database using the threshold that we devised. You can see that in 2012 we had 9,000 species, but when we applied our thresholds, we could see around 245,000 species, and for our bacteria, and 13,000 for archaea, and we had so high numbers of higher taxa detected. We detected 1,400 or nearly 1,500 phyla for bacteria and 300 for archaea. This has been somehow heavily criticized by, for example, Phil Hohenholz in his first talk, because he found that these numbers are too high. Actually, we have to understand first that the phylum has not had standing in the ICNP, so in the bacteriological code, until now. And it's the single category that has been tailored phylogenetically. So this category has been tailored traditionally as unique lineages in the tree. So Phil complained that we found too many phyla in 2014, and this probably, and because also you saw that the broadness of the variability is very high, and this is because probably different categories have been designed by different phylogeneticists. But if I scroll the GTDB and we check the GTDB data, we see that they have around 250,000 sequences of bacteria, and they find 45,000 species, and then they find 127 phyla. If we extrapolate these numbers to what would be the numbers of 2012, 1,265,000, the number of species that we would extrapolate is equivalent. So probably 2026, 246,000 are equivalent. The number of phyla that it would be found is okay, it's just the half, let's say more or less, or 3 times less, 632. But you have to understand that these data that are mostly coming from metagenomics has a different diversity thresholds. 16 sRNA surveys are probably much more diverse in the kind of samples that have been analyzed, but also the metagenomics, they only reveal the diversity in max of the most abundant organisms, and the rare biosphere is not taken into account. So probably if the complete data in metagenomics was available, this number of phyla would increase. So actually, we were not so bad in our numbering of phyla, and so I think it's not fair to disregard the usefulness of the 16S RNA in order to devise taxonomical threshold. The LTP has been evolving since 2009 until 2018 in 14 releases that you can find all of them in the Silva website. But now we move to the IMEDEA server, to my server in our institute, just for pragmatic reasons. Now you can go to the server and you can find all the downloadable data. And we have made a new release in this year because we needed to improve. So Silva has not updated his data since 2019, and we needed to make a new release, and we have been working in that release. This release that had been published this year, in early this year, I think in April, you can find a lot of novelties. First, we have a completely new, improved alignment. This is the 16S RNA alignment that you can see in the ARP package. And it increased from 43,000 to 100,000 positions because many uncultured genes, they include insertions inside that need to be taken into account. We also have been reconstructing a completely new phylogenetic tree based on this data, based on this improved alignment, and probably this is the best tree that we have now, until now, constructed using 16S RNA. This is probably the best tree that now exists, and I recommend you to check it because it is very interesting. It is the fruit of not only one treeing approach, several treeing approaches, and making a consensus tree. We have also some new fields, like we have corrected a lot of taxonomy, and we have created fields just because of trying to harmonize taxonomy. For example, we have created a field species name LPSN, but it will be changed to ICMP probably in the future, in where we give the names that are correct according to the LPSN. But, for example, we prefer to maintain the older names. So we have created a field called field status LTP, which is, for example, what you can see here, that it's a C or, excuse me, that is a C or an S. C means correct, S means synonym, and we use this because, for example, this is the case of mycoplasma. Mycoplasma subcommittee disregards the new classification, so we retain in the LTP taxonomic name the old names, but we consider and we retain in this new field the new purpose and the new proposed names. And finally, what we do, and I also recommend especially all young taxonomies, because we will not do this, we suggest changes of many taxa. We have made a list of that can be downloaded from the website, or also you can see in the manuscript. We have made a list of problematic species or problematic genera or problematic high taxa, and we have around 350 species that need to be revised, or 150 genera that need to be revised. So somebody who is interested on this can just go there and try to formulate the new classifications. We are not going to do this because it's a lot of work. So we are entering now into the era of genomics. And it seems, and in the last talks of this series, nearly nobody has been talking about 16S RNA. And this is really a shame, because what should we do? Shall we really abandon the 16S RNA? Is this the question? So do we really need to abandon this gene that has been thoroughly used? It's so perfect, it's so good for taxonomy, because now we are able to sequence genomes. I think we are still too early for this unless we are able to do whole genome with a complete set of genes, phylogenetic analysis with high power. But now we are not yet on this moment. So should we really choose between 16S RNA and genomes? And I think not. I think both are compatible and both have to be used. Because there is a general belief that whole genomes will render better phylogenies, which I really doubt. So at this moment, what we are doing is we are evaluating the three topologies of the GTTP and the LTP in order to see whether they are concordant or not. So how different or how similar are they, whether it's important to note anything. And the most surprising thing, and this is a very good message, and this is for me the best message, and probably Wolfgang Ludwig was very happy to see, because is that both topologies are surprisingly concordant. Of course, there are some cases that are to be noted, and we are now checking what is just being different. But the highly concordance is such a good news, because this means that the 16S RNA was probably the best choice to do and the best molecule to use for phylogenetic purposes. So from our point of view, there is no reason to abandon. And I think that 16S RNA has much more important advantages now in order to reconstruct phylogenies, and also especially prove the monophily premise when you want to classify organisms. And which are those advantages? Those advantages are those. For example, okay, this might see as a disadvantage for the 16S RNA, that is, it is a single essential gene, whereas in case of the GTDB, they use 120 genes that are essential. So the single gene, the 16S, represents only 0.03% of the genome if we take into account the genome mean of 3.5 gigabases. Whereas the 120 genes will be representing 3% of the genome. So yes, a bigger amount, not extremely big, but it's a bigger amount. But I will discuss this later, because the figures are not like this. The very good, one of the important or very good advantages is that we can make a unique tree for all domains which have, or all organisms having RNA. We can have a tree with bacteria, with archaea, and of course with eukarya. However, if we, as we can see in the GTDB, they are only using one tree for each domain, so they cannot really make a tree combined with both domains, for example. Another advantage is that the 16S RNA, and especially even now with the draft genomes, we have high quality sequences. So we are sure that all the sequences are there. We can see gaps that can be natural gaps, but in general, we can have a high quality supervised gene sequence with a high quality universal alignment. However, when we look at the selection of essential genes, for example, in GTDB, but it can happen in any kind of genome survey, there are groups of organisms that they may have gaps. They might miss some genes. This is just a small part of the complete data set of the essential genes of the GTDB, but you can see that there are organisms, groups of organisms, that they are missing genes, because probably this and this is one gene, probably this is another gene. And so you have a lot of gaps that are important in order to modulate the topology of the trees. But finally, when you look at the 16S RNA tree and the 16S RNA, we have an alignment, as I said, of 100,000 homologous positions. If you don't use filters, you can have 94,000 homologous positions to analyze into the phylogenetic analysis. If you put filters in order to remove homologous positions and accelerate the process of analysis, we can have just 100,000, so 1,000 1,500 nucleotides, which is 99% of the gene. And when we look at this number and we take into account a genome of 3.5 gigabases, okay, this is really nothing. So it's 0.0004% of the whole genome, but it seems that that according to all our analysis, represent the phylogeny. If we look at, for example, the GTDB and the complete alignment of the 122 genes, it's 41,000 homologous position, which is not bad. Actually, as I said, it would be 3% of the complete genome. However, the GTDB, for pragmatical purposes, reduces the alignment to 5,000 positions for the calculation of the trees. So this is actually a mean of 40 to 42 positions for each marker, if I'm not wrong. So at the end, the GTDB uses 5,000 base pairs, which is more or less equivalent to the 1,500 base pairs in order to generate trees. So altogether, what I want to say is that both approaches are equally good. I think that from my point of view, 16S RNA is much better for phylogenetic purposes and for the future of taxonomy until we are able to reconstruct phylogenies based on whole genomes. Because the 16S RNA, we have a highly curated alignment. We have the capability to reconstruct universal phylogenies, and because we have the best database ever from any kind of gene. It was the best choice and still is the best choice to use for phylogeny under my point of view. I don't discard whole genome because I think it's important to use, but as a parallel approach, but for taxonomy, I think this is the best. And so my message is actually long life to the 16S RNA, because I think it still has a long history ahead to be used. With this, I just want to thank especially my LTPG team. Especially, I want to thank especially my LTPG team.
The comparisons, one-to-one comparisons between the two. So that was really nice, and I'm sure the students especially must have understood quite a lot and very clearly as to how these things work. We've got a couple of questions. If you're ready, I can take them.
Okay, I'm ready.
Yeah. The first one was just posted in the chat box, which is from Juan Guzman. He's asking, please explain why the 16S rRNA is said to have 100,000 homologous position. Is this because of potential insertions?
Yes, exactly this. So the gene, the mean gene of all 16S RNA is probably 1,500, something like this. This is the complete gene, but there are many genes that they have insertions, and there are many genes, especially now from the metagenome surveys, if I'm not wrong, for example, the CPRs, that they have complete genes insert within the 16S RNA. So the 16S RNA is split and then has inside inserts of more than 1 kb. So this is, and when you join a universal alignment and you have to put all the different inserts in different positions, then the alignment needs to be 100,000 positions. Still, I think Wolfgang Ludwig has devised these 100,000 positions as enough to include other probably insertions.
All right. The next question comes from Tushar Loda. The question is, which region of the 16S rRNA, V1, V2, V3, V4, etc., is more appropriate to sequence in amplicon sequencing to understand the microbial community?
Yeah, well, this is a big important question, and it's difficult to decide. I mean, I didn't explain to you in this talk. I could have done, because we did a survey also in this paper of Yarza, that you could see how the information content increases when you increase the number of bases. And for me, the whole gene is the way to go. And probably in the new technologies, you will be able to sequence the whole gene. This is this is what we do. But if you have to choose only one piece, what I would choose is the one that most people uses, not the one that you think that is best. Because if you want to compare with other people, you have to use the one that is mostly used. But if I have to recommend the whole gene, excuse me.
So effectively, it depends on the limitations of the database because people are using V3, V4 for quite some time. The database is enriched with V3, V4. And that's why it's preferable to use V3, V4.
Yeah, the thing that we use, and I didn't explain to you because I don't have time here, is we use the OPU approach. So people uses OTUs or uses SAVs, but the problem is that if you have V3, V4 or V2, V3, you cannot compare really the both databases. What we do is we insert the representatives in a tree, and then we supervise the tree. And so it doesn't matter if it comes from here or from there, then you are able to see whether they belong to the same taxon.
Correct.
This is a tedious work.
Yeah. The next question comes from Somak Chaudhary. The question is, what is your opinion on the changes made to the taxonomic grouping for phyla recently that includes Proteobacteria, Firmicutes, and others? How can a researcher reconcile these iterations in names when trying to scrape information from historical literature?
Yes. Well, this is a big question, and I was not talking on nomenclature at all on purpose.
Right.
Because one thing is classification and the other thing is nomenclature. These nomenclature changes have been done in phyla, and we are discussing phyla by Aaron Oren and Garrity, and probably are extremely accurate. And extremely accurate and probably are adequate. So the other thing is whether people like it or not. You have to think that the phylum, and this is has been just now taken into account in the ICNP. This means that phylum was created by phylogeneticists and ecologists as single lineages, and they had to name them. So during all these years, this has been the Wild West in naming because anybody has given the name that they wanted, and these names have been perpetuated. And the problem is that when we include into the ICNP, and when we want to use the accurate rules, these names are wrongly formed. And if we want to use the accurate rule of the genus being the one giving the name of the phylum, then it happens this with firmicutes that will become Bacillota. So it's the procedure is accurate, and I think they did a great job. The other thing is whether for the peace of the community, it would have been better to retain the old names and recognize that those names are wrong, but we allow this just for the years and years of use. But this is my opinion. If I had been, I would have retained the old names, although I think the work of Aaron and George is probably perfect.
Right. So moving back to what LTP does, this one is from an anonymous attendee. The question is, the internal nodes in GTDB3 look like they have higher bootstrap value than the LTP.
Yeah, the LTP values bootstrap are not bootstrap for one tree. You have to see that this is the combination of many trees. It has nothing to do with the bootstrap that you know. I would recommend you to read the manuscript because there is very well explained. This was the work of Wolfgang Ludwig, and this is not the bootstrap that we know, because bootstrap it comes from one single tree and you see the very low. No, this is the combination of a multiple tree approach. So it's not the same. It's not a normal bootstrap.
Okay, right. Another question is from an anonymous attendee, and the question is,
Excuse me. And you have to think that this is a global tree. When you
Right.
want to do a real phylogeny, you have to concentrate on the branch that you want to resolve. And according
Okay.
to our experience and Wolfgang Ludwig always recommendation, you have to do different data sets, different algorithms, different filters in order to resolve the branches. You cannot rely on one tree, one bootstrap. You have to combine and
Right.
see what is the best. So
Right. Okay. This one is from anonymous attendee. And the question is, how can we make such advancement in our own phylogenetic analysis, especially while comparing organisms?
I didn't get the question. So how can we?
I think that the question is more about, sort of a take-home message. Your analysis of phylogenetic trees is very, very impressive, and the person wants to know how does the person make his own analysis so impressive like yours?
Well, you don't need to do this one analysis. I think if you can do a good phylogenetic analysis of your organisms of special interest is much better, because these big trees, they have a lot of problems because the complete data set is too big, and it's probably impossible to create an accurate single tree if you don't have the perfect alignment. So this is impressive, but this is only possible because Wolfgang Ludwig has an experience of more than 50 years of reconstructing phylogenies.
I'm actually very pleased today. Wolfgang is also actually one of the attendees today. I'm really pleased to know
Is he?
that.
Ah,
Yeah,
Wolfgang.
yeah,
Wolfgang.
yeah. He's part of the attendees today. It's very nice. Um, if he
Perhaps he
allows,
wants to say something.
I'll allow him to talk.
Yes, perhaps he wants to say something. Perhaps I said something wrong. Wolfgang.
No, I think he just left. Okay. The next question is probably my own question. I wanted to understand. We've been using 16S for almost, 4 decades now. And there have been instances, especially with taxa, that for which 16S doesn't really resolve them very well. So, what is your take home? Like, we know that some of the protein coding genes like GARase, PN, RPOD, they perform better than the 16S in terms of some comparisons for those taxa. So, how do we handle those issues?
Well, yes, I think at the end, if we want to resolve a taxonomic problem, the best thing that we can do is combine the different analyses and understand the resolution power of each of the analyses that you do. As I said, I would never discard using GTDB, for example, or whole genome analysis for any kind of purpose. But you have to be able to see where is the resolution for each of these method that you use. Of course, 16S rRNA has no resolution for some kind of groups. It happens, and then you might find genes that are better. So when you have to describe this special group of organisms, you have to just show all these discrepancies and make your taxonomic opinion. But there is no one single molecule that you can use. I think the best thing to do is to combine the different approaches. This is my
Okay.
opinion. So you have to be able to understand that different things give you different information and different resolution level.
Right.
The only thing I
So,
want to say is that
mhm,
taxonomy, this is very weird, is the only discipline in biology that relies on on opinions. The rest of
right,
disciplines rely on facts, but taxonomy on opinion. And this is very strange from my point of view.
right. So, taxonomy is the only opinion based field rather than the evidence based
Looks
field.
like, yes, it
Right.
looks like.
Wolfgang has joined us as one of the panelists. I'll take this opportunity to welcome Wolfgang. Um, I don't know if he can unmute himself. Okay. Um, did I hear something,
Wolfgang,
Wolfgang?
can you unmute, or perhaps he can't?
Okay. All right. The next question is from Chintan Bhatt. The question is, Could you please explain about conserved genome and in silico analysis of conserved genomes? What do you mean by conserved genomes? I think this question is from a beginner student, so it's very interesting for others to understand what is a conserved genome.
Conserved genome or conserved gene?
Conserved genome, I think. They're basically the core data set, the concatenated genes that we use from all organisms together.
So the question is, well, what is a conserved gene? What is the core genome? This is what,
right.
This is the question. So, well, we have different things. Well, a conserved gene is considered actually essential genes that appear in the whole group of organisms that you are working with. Core genome is more, so core are the genes that appear in all organisms of the group that you are working with. And the conserved will be those genes of the core that show the lowest rate of divergence, I guess, or mutation rates. But I think both are considered the same, no? So conserved are more or less the genes that appear in the group of organisms that you are using. Normally more universal. But,
right. Okay. Another question that comes is very interesting, and it's a confusion which many people have who are just learning the classification system. The question is: are the terminology domain and kingdom same in classification?
Well, domain and kingdom have no standing in our taxonomic system. So you can use as you want. I understand that domain was more, well, I really think that, I think they used kingdom in the beginning because the first time that Woos was using this, Wolfgang should be able to answer this, but Woos, they used first kingdom as the three kingdoms of the life, and then it changed to domain. But I'm not sure whether domain refers to prokaryotes, and I don't know. I really don't know. I use domain always. I never use kingdom.
Yeah. All right. A last question is probably something that I did not want to ask, but do you have any other gene that can compete for 16S? What do you think?
Well, probably there's 23S rRNA.
Yeah, that was one of my questions. So when many of us concatenate few gene sequences, especially doing MLST or MLSA, I've seen many papers where people combine protein coding genes with protein coding genes. Given that the substitution rates are very different, how accurate do you think are those analyses which combine both protein coding and non-protein coding genes together?
Well, this is a big question. If I had to do this, I would not do this. So and I would compare both. So non-protein coding and protein coding. It depends also on the length of a gene, on how many genes are you concatenating, because the importance of each gene will depend on the size of the concatenate, so the relevance of the gene. The difference is that if you use a 16S RNA, for example, or 23, and you have the universal alignment, you have a perfect alignment, and probably the non-coding, so the protein coding genes are just aligned using, I don't know, cluster or any other alignment, which is not manually supervised. And then you have probably a higher rate of unclear homologous positions. So I would not join both together. I would just compare. I don't know whether this is the question you had.
Yeah, I had the same question. Fanus had a comment in the chat box where he says, some of the old phyla names no longer refer to monophyletic groups. And that's probably the reason why we cannot carry forward the old names.
Yeah, this is true for proteobacteria, for example, which is not monophyletic anymore. Yeah, it's true.
There was one more question that I missed, probably because it was posted in the chat box. you mentioned that 16S rRNA gene phylogenes are better. There are conflicting groupings in 16S gene phylogeny and those of GTDB. We found that 16S rRNA gene phylogeny does not place members of genera like Klebsiella into a single group, and many others, leading to polyphyletic groupings. Now we have a situation that 16S gene is not able to differentiate species of many genera. So what is your comment on that? I think it's a similar question what I asked.
Yeah, okay. So one thing is the resolution at the species level, which it's clear that the 16S RNA has no, but probably no phylogeny based on a few genes will have. So probably you have to do whole genome analysis. And the other thing is the placement in the phylogenetic tree that is not in concordant. I have to see this, but but it's also important to know how the alignment looks like that you used, because I recommend you to check the LTP. If in the LTP it's the topology as you say, then something might have to be checked or not. But it also depends on how is your alignment, because it's very important to have an accurate alignment. This is the most important. If you don't have an alignment that is accurate, then the phylogeny that you get will be not accurate. So it's important to see how did you this researcher reconstructed history and how was the basic alignment, because GTDB, I think you just send into the GTDB and then you get back the results. But I guess that the 16S RNA has been reconstructed by himself or herself. And then it's important to see which algorithm and how the alignment was.
Okay. I think one of the last questions on the Q&A box is from an anonymous attendee. And the question is, when should we consider the secondary structure of 16S for alignment or classification purpose?
Always.
Yeah.
Always. So because the secondary structure is giving you the information on where are the correct homologous position. Always.
Right. Okay. Well, I think these were the questions we had. Wolfgang has joined us, but I don't know if he's able to hear us. Wolfgang, I'll ask one more time if you would like to make any comments.
No, I'm very glad. I'm very happy that I had been invited. For the people who are interested on the LTP, just join the subscription in the website. You just add your email address into the subscription site, because it will be very useful for you to get news and to get updated.
All right. One last question before I close. This just popped up my mind. Is the LTP database downloadable or from INDEA?
Yeah. No, the LTP, you can download it with different formats, especially in the format. I would recommend using ARC, but you can download it in any kind of FASTA format that can be used. If you don't destroy the alignment, it will be useful. So better don't realign it. So use as it is, as a master, but don't realign, because you will lose all the information on the alignment.
Right. Okay. Yeah, we are building a database of our own, so we would like to use it.
Try. Ask. You just, if you want to come to Mallorca, any of you, just join and see us and help.
Sure. Yeah. Okay. Thank you so much. Thank you, Ramon. Venju has a question.
Yes. Say last sentence, lastly, simple comments for Ramon's wonderful talk to us. Even to myself, I also learned a lot of information to me. Before, 16SR gene has been replaced the gold standard for our microbiome systematics and taxonomy for more than 40 years. And today, Roman's talk presentation gave us the conclusion. The 16SR gene still will have last long life to the world, to us.
Right.
I totally agree. But still, it's too late for you. I will maybe email to you about some other ideas in the genome, genome. We have to maybe there is some other, changes, but we still, at least the present season, we agree the CTSR gene is still the most important biomarker for us. Okay, thank you very much for your talk.
I think that the 16S has a long life still, but in the moment that we will be able to do with high power whole genome analysis, then probably will be the end of it, probably or not. But it's still
No, I think
way.
16S will live till eternity. That's my personal opinion.
I also think so.
Yeah, yeah. Thank you, Ramon. Thank you, everybody.
Thank you.
I will see you all next week with our next speaker, the last talk of 2021. Somehow it was postponed to the next year, but that will be the last talk of our first year of BISMiS Live. The talk will be given by Professor Martha Trujillo on taxonomic descriptions and reviews and in need of a new working frame. So I'll see you all in on third Saturday in the next month. And with that, we'll close. Thank you, Ramon. Thank you, Wenjun. Thank you, Ifti.
Thank you.
Thank you.